the structures, basically an introduction to all of the GI organs, from oral cavity to anus. And we're just kind of introducing the functional aspects of each of those structures. And it's really nice for you to be tying what we talk about here in lecture with what we did in lab this week. So in anatomy, we talked about the organs in the abdominal cavity, mostly the abdominal, the GI organs, so like the stomach, liver, spleen, intestines, etc. So it's really important for you to be tying that gross anatomy with more of the functional aspects and vice versa. Okay. So let's go ahead and pick up right here. And we were just beginning to get into the intestine. So we talked about the stomach, the esophagus for the most part, and we're just getting into what's happening in the intestine. So in the intestine is where mostly absorption takes place. By this point, food that you have ingested has been digested, so digestion has been completed for the most part. And here we're going to begin to move those micronutrients across the epithelial tissue of the GI wall, and that is the process of absorption. So it's going to be complete within the first 20% of the intestine length. And so the adaptations that we see of the epithelium of the small intestine are going to be suited for absorption. So we're going to see that very same brush border with the microvilli that we saw in the kidneys in the proximal tubule, right? That same epithelia that is modified for absorption. And that is going to be increasing the surface area to allow for more space for absorption. So in the small intestine, we have villi, and villi are one of the structures that increase surface area. Within villi, we have blood vessels for absorption of these nutrients, as well as lacteals. And lacteals are a connection to the lymphatic capillaries or the lymphatic system. And lacteals are where we absorb fats in particular. So fats usually get absorbed into the lymphatic system, and then through the lymphatic circulation, they make their way back into the vascular system. There's also microvilli, and microvilli are apical modifications of the cell. And so microvilli are going to be on each of the cells themselves. And I want to take a minute here to distinguish this from villi. So the villi are the outpouchings of the epithelial wall, where the cells are lining this kind of upward projection. But then on each of these cells themselves, we can see even smaller upward projections, and these are the microvilli. Okay, so they are villi, and then on top of the cells that are lining the villi, there are microvilli. The microvilli, again, we talked about it before. We should know what this is suited for. This is what forms the brush border of the small intestines. Again, modifications that are increasing the surface area. We also see crypts of Lieberkühn, and crypts of Lieberkühn are kind of similar to gastric pits, but they're basically kind of invaginations of the epithelia here, and they have cells that are lining these crypts that secrete bicarbonate-rich fluid. Now, in the small intestine, we want this bicarbonate-rich fluid to neutralize the acidity of the chyme that's coming down from the stomach. So this fluid is secreted mostly in the proximal parts of the small intestine, so mostly in the duodenum, right, the early small intestines, and then that can be reabsorbed in the distal parts of the intestine. So here's a kind of larger view of that. So here we have the small intestines in terms of the anatomy. We've got the colon, and the colon kind of encircles the small intestine, so it's going to be the periphery here. And then within here, the smaller luminal structures are the small intestines themselves. We have the duodenum, which is kind of the first part of the stomach coming down here. We've got the jejunum, which is usually pushed in the upper left corner, and then most of the ileum is closer to the lower right corner. And if we take a look closer at the inside or the epithelial lining here, we're going to see some of those modifications that we just mentioned. We're going to see the invaginations, which we call crypts of Lieberkühn, with those epithelial cells. We're going to see the villi, which are the upward projections. And then lining the villi, if we look even closer, we're going to see those microvilli. Okay, let's talk about the liver. So the liver is one of the largest exocrine glands in the body. The role of the liver is very vast. It does a whole lot of things. It helps to absorb nutrients that are coming in to the bloodstream from all of the food that we absorb. So all of the nutrients that are absorbed across the small intestine epithelium make their way into the venous circulation of these intestines, and then these venous tributaries actually drain towards the liver via the hepatic portal vein. And so the liver helps to detoxify substances from foods that we eat. It helps to process some of the nutrients, storage of some of the nutrients, right? The liver helps to store glycogen, which is one of our main energy reserves. So the liver is kind of the post office, if you will, of all the nutrients that are coming in from the food we eat, all the electrolytes and water, all of that is taken to the liver, sorted to some degree, and then the blood from the liver can be sent to other parts of the body. And so this is what the hepatic portal system is. So kind of the same gist here. We talked about this in lab in terms of the anatomy. And this is more of the functional aspect of why the blood from the GI system has to go to the liver. Okay. Let's move down now to the distal parts of the intestine, to the large intestine and the colon. The large intestine begins at the ileocecal junction. So the ileocecal junction is this border between the ileum and the cecum, which is the first part of the ascending colon. Here's where we also find the appendix, incidentally. The large intestine consists of the cecum, the colon, the rectum, and we also have a few sphincters involved here. So the ileocecal sphincter is the sphincter at the beginning of the colon, again, at that junction between ileum and colon. Now, some of the features that distinguish the large intestine are tinea coli, which are these bands of smooth muscle, these strips that run longitudinally up and down the length of the large intestine. And these help to distinguish the colon or the large intestine from the smaller intestines, which they are connected to. Now, these tinea coli actually create this gathering of the large intestine, which forms these out pouches that we call haustra. And haustra is a structure that you're probably going to see in this week's lab as well. But together, the tinea coli, which is the strip of smooth muscle, and the haustra are structures that help with the solidification of waste in the distal part of the intestine. The colon, again, now the colon and the large intestine are pretty much synonymous, right? The colon kind of makes up the large intestine. And this is where we have concentration of waste into feces, again, the solidification of that waste. We have reabsorption of most of the water here. And I should probably say absorption and not reabsorption. So we have absorption here of most of the water that's coming in from our diet. And we have that waste product being consolidated into feces and then stored until the time of defecation. So the four parts of the colon are the ascending colon here on the right. Then the colon takes this turn. We call this the hepatic flexure or the right flexure. Then we've got the transverse colon, which sits underneath the stomach pretty much. Then the colon takes a turn again to form the descending colon. This turn we call the splenic flexure. And then finally, the colon kind of wraps around as it circles the pelvis. And this S-shaped region is called the sigmoid colon. And then we get to the distal parts of the colon, which become the rectum. And then the opening itself is called the anus. Okay, so the colon empties into the rectum and finally the anus. Here we have two sphincters that are kind of guarding the emptying of feces. There's the internal anal sphincter, which is smooth muscle under the control of the autonomic nervous system. And then there's the external anal sphincter, which is skeletal muscle under our voluntary control, right? The somatic nervous system. So relaxation of both of these sphincters is required in order to open the anus and to proceed with defecation, to excrete feces, okay? So I kind of want to correlate the sphincters here with the sphincters that we saw in the esophagus, in the pharynx. So just like what we saw in the upper esophagus, where we have that sphincter made of skeletal muscle that we can voluntarily control, and just like what we saw even in the urethra, right? The sphincter that empties the urethra, that allows us to void the bladder. We also see this phenomenon here where there are two layers to the guarding. There's the internal sphincter that is controlled by the nervous system, and then there's the coordinated external sphincter that we have voluntary control of. And the coordination of both of these sphincters are required to ingest, right, to swallow, to void the bladder, and then what we see here to defecate as well. So there's that coordination between the autonomic nervous system and the somatic nervous system. So you can't have this function without both of those aspects. Okay, let's talk about the accessory glands here. So there are several glands that are helping with secretions, and that's going to be lubricating the tract. There are salivary glands that secrete saliva. There's the pancreas that secretes the pancreatic juices that have all the enzymes that are important for digestion. And then there's the acini of the liver, which help with bilious secretions that help to absorb fats. So this is what these glands look like. Most of these glands are made of the same type of cells. We call these acini. And the acini are actually epithelial cells that are lining these ducts that create whatever secretion, whether it's pancreatic juice, whether it's saliva, whether it's bile. They're all being created at these acini and then dumped into the ducts, and then they lead into the lumen of the digestive tract. Okay. Now the cells are going to be a little bit different depending on where we are, whether it's in the salivary glands, but they're basically going to transport ions. That same idea that solute movement is followed by water movement is the basis for most of the glandular secretions. So they kind of transport ions across the membrane, and then that solute transport is followed because there's now a gradient for water transport, and so water can move and create these glandular secretions. Let's talk about the characteristics of saliva. So it is going to be rich in bicarbonate. It's going to have mucus as well as enzymes. We talked about the beginning of starch breakdown, which takes place in the mouth. So it's going to have salivary amylase to aid with that starch breakdown, as well as lysozyme. Lysozyme is a part of the antibacterial property of saliva. It's going to kill bacteria, viruses, other pathogens that may be exiting the body in the oral cavity. Remember, the oral cavity is a point of entry into the body, and so this is a part of the immune system, a part of that defense mechanism. Okay, another one of the accessory glands here, the pancreas. So the pancreas actually has dual function. It has an exocrine part and an endocrine part. We talked about this before in the endocrine system last semester. So the endocrine part of the pancreas are the islets of Langerhans that we discussed, and the alpha, beta, delta cells that are found there. The exocrine part of the pancreas is pretty much everything else. So while the endocrine are these aggregates of cells, all of the rest of the pancreatic tissue is functioning as the exocrine part of the gland. The functions here are going to be secreting that rich bicarbonate fluid that's going to neutralize the chyme into the duodenum. It's also going to secrete lots of enzymes. They're going to have amylases, lipases, proteases, nucleases that are breaking down starch, fats, proteins, and nucleic acids, respectively. So the fluid that's coming out from the pancreas is rich with enzymes, and relative to its weight and its size, there's no other gland that secretes as much protein as the pancreas does. Remember, enzymes are proteins themselves, right? So as far as the work of the pancreas, there's no other gland that matches how much protein this gland actually secretes. And we can see the connection of the biliary tree to the pancreas. So remember, the biliary tree is releasing bile into the duodenum, whereas the pancreas is releasing all of that, that enzymatic fluid into the duodenum as well. And if we were to look even closer at the pancreatic tissue, we would see those same acini, right? The acinar cells that are lining these ducts that are making these enzymatic secretions, releasing them into the ducts, and then the ducts will eventually flow through the pancreas and make its way onto the lumen of the GI. Okay, we'll talk about the liver here some more as far as its role in bile secretion. So the liver does a lot of things. We mentioned this earlier. It's going to store glucose to glycogen. It's going to process amino acids into fatty acids. That is a process called gluconeogenesis. It's going to package and store triglycerides and cholesterol, as well as lipoprotein synthesis. So lipoproteins are required in order to transport fats around the body. And so the liver has a lot of functions. Continuing on here, it's going to remove old red blood cells. So you would have talked about this when you looked at the process of erythrocyte production and erythrocyte destruction. So as you would have studied, the old dying erythrocytes are transported to the liver. They're broken down. That hemoglobin is liberated. Bilirubin is made and secreted out onto the body, and that hemoglobin can be recycled to make new erythrocytes. So the liver helps in that process of catabolizing hemoglobin. It's going to help in elimination of waste and toxins, again in the form of bilirubin, which is the bile form or the bile component of those old erythrocytes that can be eliminated from the body. It's going to help in the synthesis of plasma proteins. So all of those clotting factors that you would have talked about as a part of the clotting cascade that are secreted into the blood in their inactivated form, all of those factors are secreted from the liver. And then finally here, it's going to help in the secretion and modification of hormones. Lots of different hormones are made in the liver. We also have albumin, which is one of the most important hormones made in the liver as well. Let's talk about the biliary system. So the biliary system or the biliary tree is comprised of the liver, the gallbladder where bile is stored, and then the associated ducts that transport that bile into the duodenum. So the liver makes bile, as we know. The gallbladder stores that bile until the time of release. And then the common bile duct is the duct that transports that stored bile into the duodenum. And so the premise here is that we'll talk about what bile does later on, but the premise here is that bile is always being made, but it's not always being used. And so until it needs to be used, it is stored in the gallbladder. This is where we have the cystic duct. So the cystic duct has this retrograde transport where it takes this made bile and stores it in the liver. And then when we have a fatty meal, the gallbladder can begin contracting, release that stored bile, and it can make its way via the common bile duct to the duodenum. We also have a few sphincters here that help to guard these openings so that bile is not always being, released into the duodenum. It's called the sphincter of Oddi, and it's going to regulate the bilious secretion from the gallbladder, but also the pancreatic opening with those enzymatic secretions as well. So the sphincter of Oddi is guarding the opening that's coming from the pancreas, which is the major pancreatic duct here, and then the common bile duct, which is coming from the gallbladder. Briefly here, we'll talk about the liver anatomy. So the liver, because it's one giant gland, it's made of these acini. And these acini are basically creating the bilious secretion and flowing that towards the hepatocytes. So there are these capillaries called sinusoids that are forming the liver, and the sinusoids are simply capillaries with really large holes in them. And so the hepatocytes, which are the liver cells, take up the products to make bile via these sinusoids and secrete this bile onto canaliculi. We've heard of the term canaliculi before. It's really just a small duct. When we talk about the lacrimal canaliculi, it's a similar type of structure, a very tiny duct. And so these canaliculi uptake this bile, release it onto the opposite side of sinusoids, and then it can drain into the bile ducts. From there, it'll go to the hepatic ducts, so the right and left hepatic ducts respectively, then into the common bile ducts, and then it can make its way into, excuse me, not the common bile duct, the common hepatic duct, and then it can make its way into the gallbladder, and then from the gallbladder, it'll go to the common bile duct, and then to the duodenum. Okay, so the flow is going to be right and left hepatic duct, common hepatic duct, cystic duct, and then common bile duct. Okay, so here's a really nice study table. This is going to just show all the organs, all the structures we talked about, and then an overview of their function. So this is going to be a nice reference table for you to study. You can add some properties that may be left out here and use this as a good summary for everything we talked about in this